It's 7:30 now. Hello again, guys and girls. Biology is most likely one of the underrated topics on this year's camp. So I'm very glad to have a speaker here who will give more insight into that. Gene hacking. Please give a warm applause to Mark Jewell.
Thank you. So I'm going to talk to you about hacking DNA. Most of you are probably familiar with programming or electronics, but we're beginning to hack DNA or hack biology. I'm going to give you a bit of an overview on the technologies involved. So first of all, why would we want to hack biology? Well, biology is a very different system from the ones we usually work with, but it has some nice properties, such as self-replication. Biology has a nice property that if we program one cell, it will self-replicate. We can build microscale devices. This is a virus that attacks bacteria, bacteriophage. It's 200 nanometers or a little bit less. And this is one of the component parts that goes inside of a cell, 2.4 nanometers. So you could say this is a kind of nanotechnology, though the devices themselves are microscale, the components are often nanoscale. And we can use it to build macroscale devices. We see this in nature. We're not quite there yet with the technology, but we know the capability is there. Biology, you can use the environment itself as the building material. This tree, 90% of its dry mass is from CO2, carbon and oxygen from CO2. So you can build quite large devices from just the gases surrounding you. We can produce some interesting chemicals, or nature can produce some interesting chemicals. This is a wormwood, type of wormwood, and this plant contains artemisinin, which is an antimalarial drug. DNA has a nice property that it has a standardized language. This is something we don't have in computer science. We have a genetic code composed of 4 letters, and we have 20 different amino acids that these 4 letters can specify. And this is true for all biology. Biology gives us the power to hack ourselves. If we master biology, we'll be able to master the technology that built us. And just to get a bit of a space angle on all of this, if we can hack ourselves, then maybe we can solve some of these problems that NASA has identified that comes with space travel. So we have cardiovascular deconditioning, decreased immune function, bone loss, some bad stuff that we probably will have to deal with if we want to stay in space for a long time. Okay, what's the state of the art of biohacking or synthetic biology? Synthetic biology is the technology of engineering biology. Well, we can produce chemicals in different organisms than the ones that they're normally produced in. This is an article about the production of the artemisinic acid, which is a precursor for the artemisinin I showed earlier. And these guys out of UC Berkeley, they managed to produce this drug in normal beer yeast or baker's yeast. And by doing this, they're able to produce it much more efficiently and for a much smaller cost. So malaria is a huge problem. It causes 20% of childhood deaths in Africa. That's about 2,000 deaths per day. And these guys produced artemisinin in yeast, which is lowering the price, and it's also going to solve some problems such as availability. Sometimes you have a low crop yield or you have market fluctuations, and that can lead to problems with availability. And this drug was created in 2006. And the proof of concept, not the drug, the proof of concept about producing a drug in yeast, and it'll hit the market in 2012. So it still takes us a while to do these kinds of things, but they're possible. This is a student project. that I think is pretty cool. It's called E. coli, and they took an E. coli bacteria and they modified it to produce all these different colors. And this is very useful because if we can produce different colors in bacteria, then you can hook those color-producing elements up to, let's say, sensors or something you're interested in, and then you can get an output that's very easy to read out. So instead of having to go and do very difficult analysis in a lab and have very difficult to use and expensive equipment, if you can have the bacteria produce the color directly based on the inputs that it detects at a micro scale, then you could imagine all these nice uses, diagnostic uses, and this is what they imagined. So this is an imagined application of B. chromai whereby it would live in your gut and it would have detectors for all the things that make you sick in different ways. And based on this, it would produce different colors, so you could very easily see if something's wrong with you, and not just something, but have an idea what it is. This is another student project. This is an E. coli that was modified to detect nitrogen in soil. And nitrogen in soil is, of course, very important for production of food crops. And if you know something about how much nitrogen is in your soil, then you know how much fertilizer you need to use, and maybe something about what kind of crops you should grow this year. And they made these little gel beads with these E. coli bacteria that you just spread over a field, and they'll light up based on the amount of nitrogen. And then you can go out at night and you can have a look and see how much it's lighting up when you put a black light on it. You can make cancer-killing bacteria. This is a project by Anderson out of UC Berkeley, and these bacteria were modified to detect when they're near cancer and detect when there are enough of them near cancer, and then actually invade the cancer and start killing it. And the idea is you would put this into your bloodstream, these bacteria that have been modified to help you instead of kill you, and they would find the cancer, and once enough of them find the cancer, they just destroy themselves and the cancer with them. Not quite at a stage where you can use it yet. One of the problems with this technology is that our immune system is really good, so it has a tendency to kill off bacteria that's floating around in the bloodstream. People are starting to produce some interesting polymers, like spider silk, which is a really strong material we'd like to mass produce. The problem is that spiders, they don't like each other very much. They tend to destroy each other if you try to farm them in a big area. And it's very difficult to farm spiders if you have to keep them separate with their own little area and harvest the silk. So if you can produce spider silk in an E. coli bacteria, then you just have a big vat of E. coli growing, and then you can grow them for a while, break them down, and then extract the spider silk. Some people work on this project. It's still not quite up to the spec of normal spider silk, but it's getting there. And all these projects were done with two organisms, the two best characterized, most well understood organisms in biology, E. coli and normal beer yeast. So we're at a very early stage right now where we only have the capability of engineering some of these very well understood organisms, or at least it's difficult to use the more interesting organisms with the multicellular organisms. But these two we know pretty well, and we can do a lot of stuff with them right now. Okay, so we've established all the cool stuff you can do with DNA, but how do you actually do it? How do you hack DNA? Well, when I look at something I want to hack, I usually see if there's any documentation. It's a nice first step. Then I want to figure out how do I compile it, if it's some kind of code, and how do I run the code? If you know this, then you can hack any system, right? So the problem is that we didn't really get a lot of documentation from nature. Evolution is not big on code comments, but we have some nice textbooks where all of the science we have to this date can be looked up very easily. And you can go and find some review articles that show you how a lot of stuff in biology works. Of course, you can go to this nice site called PubMed, and you can find articles about almost anything in biology. even though we have a lot of articles and a lot of information that we've built up, and we've built all of this up by reverse engineering biology, we're still at a stage where we don't know a lot. E. coli, the most well-understood organism, we only know about 3/4 of the components, what they do, and about only 2/3 of the components have been characterized and experimentally verified, so we're sure what they do. And of course, even though you might know all the components and what all the components do, you still have to figure out what do they do when they're working together. And this is very difficult to simulate. It's a very stochastic environment, a lot of stuff interacting in different ways that it wouldn't if it had been a designed system, but this is an evolved system, so it's fairly difficult for humans to understand. Okay, before getting into how to write the code, I'm going to talk about some of these technologies that are the basis of biohacking. There's sequencing, synthesis, assembly, and PCR. And sequencing is the act of taking some DNA or an organism and getting its DNA and then reading it into a computer so you can look at it. Synthesis is the opposite. It's like DNA printing. So you have some DNA information on your computer and you want to turn it into an actual DNA strand, you use a DNA synthesis device. Then, because synthesis is very expensive right now, it's getting cheaper all the time, but it's still pretty expensive, you might have some DNA lying around for different components. So you need DNA assembly to stitch together pieces of DNA into bigger systems, and you need PCR for grabbing a piece of DNA out of a bigger piece and amplifying it. So if you have a little bit of DNA, you can use PCR to amplify it and get a lot of DNA, but you can also use it to pick out a specific piece of interest and then amplify only that piece. I'm just going to give a quick overview of the central dogma of biology. And this is, once we understood this, we basically understood the principle of how all biology works, so it's pretty important. You have some cells. This is E. coli magnified 15,000 times, scale of 2 micrometers. This line is 2 micrometers. And inside of these cells, there's some DNA. This is green fluorescent protein. It's a nice protein that lights up green when you put a black light on it, and this is the DNA code for it. 720 base pairs, or nucleotides. What happens with its DNA inside the cell is that it's transcribed, is the word, it's turned into RNA when it needs to be used by the cell. So you have this double-stranded DNA that we've all seen before, and it's turned into single-stranded RNA. And this is kind of the working copy that the cell uses, whereas the DNA is the archive copy that it keeps around when it's multiplying. The RNA is then turned into this string of amino acids, which is also known as a protein. So for each of these 3 letters, for each 3 letters in DNA, they're read out as one block, and that one block codes for one amino acid, which are the base components of proteins. So you go from DNA, from a string of DNA to a string of amino acids, where you get a third the amount of amino acids of the DNA. And these little three nucleotide blocks are called codons. So if we have to look at this from a computer science perspective, you have nucleotides that are four different kinds of nucleotides, so that's 2 bits of information right here. And then for a codon, you have 3 of these, so that's 6 bits for one amino acid. Oh, and I have my slide here that shows that. So you go DNA, RNA, amino acid strand, and just as the bases in DNA have nice letters, the amino acids also have letters, so you can have an amino acid code like this. And then once you have an amino acid strand, what happens is that it folds into a structure. And this is actually one strand of amino acids that's folded into the structure of the green fluorescent protein. It's the structure that gives it function, the structure and the different amino acids and the different positions. So yeah, proteins have function. That's why they're useful. So what I just showed you tells you how all the components or most of the components in living organisms are made. You You have DNA that's turned into proteins. There's also some other stuff. There's metabolites, stuff modified by the proteins, but the actual machinery, most of it is proteins. And these proteins, they have different kinds of function. There's the green fluorescent protein we've already seen. There's something like insulin, and green fluorescent protein is very simple. It just lights up. Insulin actually goes outside of the cell and to other cells, and it can signal other cells in different parts of the human body. So you have some proteins that function to signal. You have signal receptors. So inside of cells, if you can imagine this, we're looking at the edge of the cell wall, and this is inside the cell down here, and up there it's outside the cell. There's some proteins that actually sit at the cell wall, and they detect signals, and they change conformation inside, and they can send the signal to other proteins inside of the cell. There's enzymes that can modify chemicals in different ways. We say they catalyze chemical reactions, so we have enzymes that can change the reaction rate of the reaction that would normally take place at statistically insignificant rates. So these enzymes can make sure that different chemical modifications happen at very fast rates inside of cells, and that's why cells can produce a lot of interesting metabolites. So E. coli has approximately 4,500 proteins or parts. And as I said before, we don't know the function of all of them, but we know a lot of them. So now that we know that we can have these functional units based on DNA, how do we write DNA to change the function? Well, one way would be to write new components, write the DNA for completely new proteins. We're not very good at that yet. There's some initial research where they've created completely novel proteins, but what's happening mostly right now is that we discover proteins in nature, and then we find the DNA sequence for them, and then we can use those proteins in new combinations. So we use libraries of pre-built components to build new systems. And this is an example of a library of biological parts. This is the Registry of Standard Biological Parts. It's a completely open site where you can go and you can find parts for all different kinds of functions that you're interested in. And you can get the DNA sequence. You can order the DNA as an actual physical piece of DNA and get it in the mail. And you can combine them in different ways. So the problem with these devices is that they don't have this. They don't have a data sheet like we see in electronics, because if you have a part, you're really interested in knowing exactly how it works under different circumstances and knowing when it breaks down, what the linear range is, what kind of input and output it has. And a lot of the components in the parts registry, there are a lot of components there, but a lot of them, they're not very well characterized. And there hasn't been a really good standard for characterizing these parts. So it can be difficult to use. It's kind of like going into a library of open source projects, and some of them will be very undocumented and some of them won't be, but most of them here pretty undocumented. Most of them contributed by students working on a deadline. So this is an example of what an electronic data sheet could look like for biology, or an equivalent. And this is what we would like to see for all the parts in biology. So there's an organization called BioFab in California right now that's trying to create this kind of library of professionally characterized parts. And you can go online to the website and you can find different parts in your library, and you can find a part that has the exact function you want. You can look at the data. So this is we're beginning to get well-characterized parts. Okay, once you have these characterized parts, how do we write programs? Well, in computer science, we do this for a Hello World program. In biology, it's a little bit more difficult. You need a few more parts to do a Hello World. I'm not going to go into the details of these parts, but basically you need something to kickstart the transcription from DNA to RNA, and then you need another part to go take it from RNA into the protein, And then you also need a terminator to show where the RNA should end. So this is the gene, this is the part. You also need some other control sequences around it. And these control sequences, they specify how much of the part is made, and they can also specify when the part is made. So this is just a basic hello world that always outputs green fluorescent protein, but you can imagine more complex circuits that only produce it when something happens, such as the nitrogen detection system I showed earlier, that detects an outside signal and then it produces GFP in response. Okay, well, how do we compile and run the code? Well, there's no real equivalent to compiling in DNA because we're still working with the base code. We're still working at the machine level, machine code level. We don't really have abstract programming languages yet. So we have to take these parts and combine them, and that's called assembling the parts. And you can get the parts in different ways. You can synthesize a part, you can pay a company, go online, put your credit card info, put the DNA, they'll send you back the DNA, and then you can assemble them. You need some chemistry, you can buy kits for this, assembling different parts into a single strand. Then you need some way of putting it into a live cell. And usually what you do, or a simple way of doing this, is to use a circular piece of DNA called a plasmid. So you take the DNA you assembled, you put it onto the circular piece of DNA, and the circular piece of DNA has all the stuff you need to express the DNA inside a cell. And you put the plasmid into a living cell, and then when you grow the cell, all the DNA and the chromosomes of the organism will be expressed, but also the circular little piece of DNA that you put in there. So you can have this little, not an extra chromosome, but a little extra piece of DNA that gets replicated with the whole system. And you put this plasmid into living cells by, you could do it in different ways, but it's surprisingly easy. You can use something called a gene gun for plants that has these little gold particles that are charged and the DNA attaches and you can shoot it in, and some of them will end up in cells and they'll replicate and so on. You can also just use some chemistry to stress the cells out. If you have some E. coli, you can put them in some chemicals where they get kind of not broken up, but they get kind of stressed out, and then they start taking up DNA. You can also give them a shock, and that does the same thing. So it's pretty easy to get your DNA into the cells once you have it on a plasmid. And of course, growing cells depends on what kind of cells you have. For E. coli, you can grow them, and you basically just need some sugar and some base nutrients to grow them on. Okay, so one of the challenges that we have, apart from the characterization problem, is the lack of modularity of parts. The biological systems are not designed systems, and that means that there's no nice modularity where parts don't interact in ways you don't want them to. All the parts interact, and not all the parts, but a lot of parts interact in many different ways that are very difficult to characterize. And all this is kind of floating around in this big soup of parts. And so it's very difficult to stop things from interacting in ways that you don't want. There's a lot of effort going on right now to try to make more modular parts, but it's yet to be shown if we can really have completely modularity in biology. But if we could get this modularity of parts, of course, it would be very easy to simulate, and it would be very easy to design new systems, because then you know, hey, this part has this input and this output, and this other part has another input and output, and you hook them up and you get this result. Right now, often when you try that, you have to take into account all the other stuff that's also floating around in the cell, and all these interactions that you probably didn't think of. So it's fairly hard to do biology, to do synthetic biology. But we have some pretty interesting systems built. In E. coli, someone implemented a genetic toggle switch. So that's kind of like a flip-flop circuit. And people have implemented these logic gates in cells as well, and ways of communicating between cells, between logic systems in different cells. So we do have the beginnings of something you call abstractions on top of biology. We have the things we've used in electronics to build abstractions, at least. So maybe we can build more abstractions. So what we'd like to have, or what I'd like to have, is a more abstract language, a biological programming language, where I don't have to take parts and put them together and simulate and try it in biology and tweak it if it doesn't work. I'd like to have an abstract language where I can just write some code and click compile, and it'll find the parts, put them together, and then give me the result I need. There are some efforts going on right now out of BBN laboratories, specifically at the east coast of USA. But it's very early work, and even though we have some initial implementations of this, it doesn't really compile down to working biology yet. Hopefully it will soon. Okay, well, now I'm going to talk a little bit about biohacking in the do-yourself bio community. All the stuff we saw before, a lot of it was by students, but it was all done in institutions, in universities or research institutions. So I'm going to show you some of the stuff you can do if you don't have a lot of money and if you're just working out of your garage. There's a whole community right now, and you can go to doyourselfbio.org and you can see all the different places in the world where people are working on, or people have spaces where you can go and work on biology. Some of these are just groups that meet up every once in a while. Some of them actually have physical labs where you can go and you can have workshops and learn how to hack biology. And some of them are pretty advanced. This is GenSpace in New York. And GenSpace actually have their own standalone lab where they do workshops, where they have some interesting projects going on. They're actually doing synthetic biology. They're actually modifying circuits, and this is their own little PCR machine, the DNA amplification device I talked about. So they actually built one themselves. And basically it works by heating up and cooling down some different enzymes and some DNA and some building block materials. So that's a really useful device to be able to build. And this is another interesting project. They're building this balloon sampling station that you send up into the atmosphere, and then it will sample some of the biology that's floating around up there. And mostly they're doing this because, well, who knows what's up there? No one really knows what's floating around up in near the stratosphere. If there's any life up there, it would be interesting to find out. So they're going to grab this, use the sampling station, grab some of that if it's there, take it down, have a look at what kind of biology they get, and they could do that in multiple ways. They could try to grow it, but maybe it won't grow because it's up there where there's no oxygen. They could also try to sequence the DNA if they can get some people with a sequencing machine to help them out. This is the BOSS lab in Boston, and they have a little space in a hackerspace called Sprout. And they're really cool because they have this extreme transparency. You can go on their website and you can look at all their lab notes. So everything that they do is logged. You can see everything they do, even if it fails, even if they do something stupid like spill a chemical they probably shouldn't have spilled. It's there on their website. And this is a really important thing to have because I'm gonna get into this later, but there's some people who are a little bit worried about these garage biologists just working on synthetic systems in their little garage spaces. So it's important to be very transparent and open about what we do. This is my own little space that I co-founded with some people at the Copenhagen hackerspace called Labitat. And a couple of projects here is one guy did a colony count application for Android. So when you grow bacteria, a lot of the time you grow them on these plates, and you want to figure out how much grew, because that's a measure of how much your bacteria are thriving. If they're not growing very well, you don't see a lot of colonies. So this is an application where you take a photo and it assists you in counting the colonies really quickly and then doing some calculations to figure out the concentration and how well they're living. We also have this safe for dermalfuge, so I'll get to the dermalfuge later, but basically it's a home-built centrifuge that was improved to not kill you. There's a little bio group at Noisebridge, and we made this little thing called the BioBoard. It's a prototype right now. We're working to make it better. And the BioBoard is interesting. It's not really synthetic biology like I talked about so far, but this is kind of a different angle on biology. Of course, you don't have to genetically modify biology to do biohacking or to do-it-yourself biology. A lot of people, they just want to analyze biology and figure out maybe how to make some better beer or brew some nicer kombucha, something like that. So at Noisebridge, we're trying to quantify the brewing process of kombucha, and we made this bioboard with a little optical density sensor that gives you some kind of indication of how many cells you have. And this is a pH probe we bought from an aquarium and built an amplifier for it, so you can do a little pH measurement. And this is the prototype of an uptoad to figure out how much oxygen, dissolved oxygen you have in the kombucha. Of course, you can use it for all kinds of different things, but we're hoping to make a big set of components, set of measuring devices, so you can quantify any kind of bioreactor, whether you're brewing beer, kombucha, or making a cure for malaria. LA Biohackers has a really cool project. So there's this problem when you have a lot of crops you want to grow, you need a lot of ammonia to feed the crops. You need a lot of fertilizer, and that costs a lot of money. About 1% to 2% of energy in the world is spent on this nitrogen or ammonia production. And if we can figure out a better way of doing this, then we can, save 1% to 2% of the world's energy, maybe. Some plants, they're really nice because they can get ammonia from the atmosphere. They can take nitrogen and they can fix it to ammonia themselves inside of these little nodules in the roots. But only some plants do this, and there's a problem with the enzymes that are responsible for this in that they don't like oxygen. So the plants that do this have this really intricate system where they prevent oxygen from getting in to where the nitrogen is made into, is fixed to ammonia. And we'd like to put this enzyme, or LA Biohackers would like to put this enzyme into every plant that we grow as a crop, because then we don't need to use fertilizer or as much fertilizer. So they have this experiment going with where they're trying to grow this weird strain that someone found that actually can do the nitrogen fixing with oxygen present. Problem is that it only works at 65 degrees. So they're trying to see if they can change that and modify it to work at the temperatures that normal plants grow at. If they're successful in this, then they'll be able to save us a lot of energy and a lot of fertilizer use. And this picture here is their little incubator for this very exotic organism that needs a lot of hydrogen, needs some hot carbon dioxide and a little bit of oxygen. There's a new facility being built right now, a biohacker space called BioCurious, and they just got their facility, so they haven't opened yet, but they're opening soon. 222 square meter facility in Mountain View. And they got Kickstarter funding, $30,000 of Kickstarter funding, so they actually have all the equipment ready. They're moving in, and hopefully they'll open soon. All these cool projects people are doing are not limited to just the do-it-yourself biospaces. There's collaboration between the different spaces. In GenSpace, there's a group that participates in this yearly competition of biology called iGEM. And they're working from the space, from the hackerspace, instead of from the biohacker space, instead of from the university, even though they're participating in a scientific contest. LA Biohackers has set up initial collaboration with the place called BGI, which is the largest DNA sequencing facility located in Beijing in China. And it's very good to see that these institutions are open to collaborations with do-it-yourself biologists. This also shows that we're taking it very seriously, that people are aware that you can actually contribute something that's useful just out of your garage or your little hackerspace. So we still have some challenges with these do-it-yourself or biohacking projects. There's a problem with getting the materials. We have some different materials you really need in order to do biology in the lab. You don't need materials for everything. A lot of biology can be done just using a computer, bioinformatics, analyzing data, building computer models. But if you want to actually modify DNA or grow biology, you need some equipment, some chemicals, some biology. That can be difficult to get. Some companies will send you the chemicals and biology if you're at a commercial address, and that's the only requirement. But it really depends on your country and where you live, what kind of zoning there is on the place where you live. So depending on your country's local regulations, that might be very difficult or very easy. Equipment is really expensive. but a lot of the equipment you can build yourself. You can get kits. I'll show you some of them later on. Some of the equipment you can buy on eBay, of course. There's a lot of biotech companies that went belly up recently. So a lot of this is for sale on eBay. There's also some challenges with laws and regulations, safety and ethics, copyright and patents, and I'll get into this now. Oh, let me just show you some of the equipment here. These are some of the projects, open hardware projects for biotech. So this is the Open PCR project. It's a completely open kit for amplifying DNA, and you can get it for $512. Buy the kit, assemble it, all the software is included, then you can start amplifying DNA. You can buy some of the primers online, these little pieces of DNA you need to only amplify a certain part of DNA. So you can start doing some pretty cool biology just having this device. Then if you combine it with this device, this is an electrophoresis box that lets you separate different pieces, DNA of different sizes or proteins of different sizes by dragging it through this gel. So you apply some power, it drags the DNA put in it across a gel, and then it separates by size and by charge of DNA or proteins. Very simple device. There's an open kit you can buy as well. This is the Dremel view just showed you earlier. Basically, it's a 3D printed head for a Dremel, and you can put your little containers in it, and you can spin it up to about 30,000 RPMs. You probably shouldn't, but you could. And that also can separate out proteins of different sizes in a very simple manner. So we have a lot of do-it-yourself equipment being built, and combined with stuff you can get on eBay, you can actually set up a lab in your own home pretty easily. But what about the laws? There are two different regimes I've looked at, the European Union and the US, and they're very different. In the European Union, we have all these European Union level directives that all the countries have to implement. So there's some directives that are kind of general rules about what you can do, and then there's the local implementations of those rules that differ from country to country. So there's a certain minimum, and then they can make it more strict. And even though the local country implementations vary, it basically says that the European Union level directives basically say that you can't do genetically modified organisms, at least not in the methods that we usually use, or using the methods we usually use, without getting a certification, without getting licensed to do it. And that's true for all European Union countries, so that makes it really difficult to do genetic modification in the European Union. There's a lot of other cool biotech you can do, but it makes it kind of difficult to get started, and it makes it really, really important to have these communal labs that get certified so that people can come there and use a certified lab and not be breaking the law when they do genetic modification. So right now there's only one do-it-yourself biolab that's been licensed in the European Union that I know of, at least, and that happened last month. That was Cathal Garvey in Ireland, who got his own little lab certified, and he's now holding workshops where you can go and without any genetic modification. In the United States, it's different. They're very different. There's a lot of regulations, but they don't really apply for do-it-yourself biologists. And the reason they don't apply is that there are regulations for people who get funding from different sources. So if you're not getting funding from anything, and you're not selling your product on the market or doing anything like that, and you're just kind of experimenting on your own, then there's no real laws about GMO in the US. You can just go and do it. But basically you can go and do biology. But that also means that, well, what if that makes it a little bit dangerous, because there's a bigger chance that people might freak out that you're doing all this dangerous stuff, this genetic modification, without any sort of certification or anyone looking at what you're doing. And the communities in the US are very aware of this problem. And what they've done is basically they've gone and talked to the FBI and made sure that every time they do something, the FBI knows about it. And the FBI is very friendly, and they'll have meetings with you if you want to talk to them about what kind of requirements they have and what kind of reporting they're expecting. But basically, no regulations, just make sure your local law enforcement and the FBI know what you're doing, or at least the FBI. There was a presidential commission for the study of bioethical issues that recently published a report, And the conclusions for do-it-yourself biology was that it's no real danger yet. It's still too difficult to do anything really dangerous, or at least build novel organisms that are really dangerous. And the recommendation was that there should be active communication between law enforcement and the do-it-yourself groups. And maybe in the future we can talk about constraints, but right now, fairly positive report. And then the FBI invited everyone to a workshop. Everyone who was active in do-it-yourself bio, I think, got an invite with all expenses paid to Washington, D.C. to meet with the FBI about biosafety and do-it-yourself bio. And I think most people, when they saw that, they didn't really know what to think. But basically what they said was they told us how to report if there's a problem, who to go to, how the internal FBI structures are organized, and all this kind of stuff, and just to keep everyone in the loop so that there's not an incident of local law enforcement freaking out about something and then escalating, so that the FBI knows everything that's going on and knows that you're not a bioterrorist with a secret lab. Okay. So there is a problem of both this ethics and public perception of biology. And that's something that, of course, everyone is doing do-it-yourself bio is very aware of. So there is 2 workshops to form this kind of code for ethics code for do-it-yourself bio. One workshop was in London, another in New York. San Francisco. And in these workshops, we tried to sit down and figure out a code of ethics that we can all agree on. Figure out what kind of things do we stand for, what kind of things do we not want to involve ourselves with, and to kind of have this sign that we can hold up and show the media and show everyone that we're responsible and we've thought about safety and ethics. And we're still working to compile this into a list that we can agree on. And this will become some kind of minimum list that a lot of these spaces, hopefully all of them, will adopt. Kind of like the Hippocratic Oath for doctors, but for do-it-yourself biologists, hopefully. And then I think we'll see a lot of variations on this code because it's difficult to get a bunch of hackers to agree on anything completely. So when we have this established code, you'll probably see it pop up on all the do-it-yourself bio websites for the different groups in slightly different implementations. Okay, one last challenge: copyrights and patents. So we all know how challenging this is in software, and in biology, it's different, but no less challenging. Let me just talk a bit about copyright. So copyright basically doesn't apply to biology. That makes it really difficult because we can't use any of the licensing schemes we've seen. We can't use GPL or BSD license or anything like that. And the reason for this is that since we're not, the interpretation right now seems to be that we're not building proteins from scratch. We're finding them out in nature and reusing them and modifying them. So what we do isn't seen as a creative work. And that's why copyright doesn't apply. And that seems to hold true for not just the US and European Union, but international copyright law in general. So basically, we have to get to a point where we can create something from scratch and have it be a creative work before we can use these nice open source licensing schemes. Now, patents is really the intellectual property that's used for genes. There's a lot of patents on genes and genetic systems, and that's what you have to deal with in biology. So there's been an attempt by Drew Endy out of Stanford to make this public agreement, and it's an attempt to do something like an open source license or a free software license, but for biology, given that we don't have copyright. And it's kind of weird when you're used to these normal software licenses, in that you have 2 agreements, and not only does the person who makes the stuff available under this license have to sign, the person who uses it has to sign as well. So you have this document you actually have to sign and send in before you can start using this BioBrick public agreement. BioBrick, by the way, is a reference to the standard that's used on a lot of the parts that are freely available online. It's a standard that makes it really easy to take different parts and assemble them. And this license was made with these parts in mind. So, as you can see here, there's some stuff you agree to, and it's the same kind of stuff that you see in open source licenses, but there's also this will not assert patents, and because there's no copyright, that's kind of the same as saying you can freely use it. And there's also this nice disclosure of applicable patents. The thing is that it, of course, has to say you disclose the patents that you know about. Because what happens if you say, hey, there's no patents on this, feel free to use it, and someone comes along with a patent you didn't know about. So you can't ever really be completely sure that no one's going to sue you, but hey, it's the same in software. So yeah, contributor agreement, user agreement, it's very simple. Basically just says you've read and understood what the contributor signed, and you're not going to use it for harmful things. That's a little bit different from open source. And then attribution, and you're not allowed to remove these biological tags, because then you can't really trace it back to who made it. Okay, so given all this, how do you get started with biotechnology, hacking biotechnology? Well, it really depends what you want to do. It's very easy, like I said before, to do dry lab only stuff. In biology, we talk about dry lab versus wet lab. Wet lab is where you're actually using biology and chemicals. Dry lab's just on the computer. So dry lab only, very easy. Get a computer, start downloading stuff, and lots of sequence information available. You can analyze, lots of resources online. Most programming languages have some kind of library for dealing with biological information. Then if you're doing non-GMO biology, let's say you're analyzing kombucha, or you're trying to figure out what kind of proteins are in something, and you're not actually modifying anything, also fairly easy. It might be difficult if you need some kind of specific organism you can't get from nature, you need it sent to you. You might still be subject to some different rules and regulations. But also, non-GMO biology, it's really only very difficult when you want to do really interesting stuff where you're building your own genetic circuits and implementing them and trying them out. Right now, I can only say that if you really want to do this, either move to the US or get registered, get certified, figure out what the local process is for getting certified, and start it, because it's going to take a while. But hopefully we'll see spaces, kind of like the hackerspaces, but biohackerspaces, certified, popping up around Europe in the next couple of years. Yeah, so really good resources for getting started. Biology textbooks, you can get them cheap. It might sound boring to start reading a textbook, but they're not that difficult to read. They're not full of a lot of difficult math or any really difficult to understand concepts. You can just kind of pick one up, start reading about how molecular biology works. Open access articles. There's a lot of articles now that are open access, especially in synthetic biology. So most of the interesting articles right now, you can just go and download them for free. Of course, if you have university access, you get a lot more. You can access all the Nature articles and Science articles, but there's more and more of these journals popping up, kind of like there's one called Public Library of Science 1 that has a lot of interesting articles that you could go and read. And of course, you can just start growing things. That people have been doing this for many years, but maybe you could start growing things and then analyzing them instead of just blindly growing and hoping it works out. We're hoping the BioBoard will help with this. And like I said, you can download data. You can download complete genomes that have been sequenced from many different organisms. You can download like yeast, human, E. coli, corn. Just go to the NCBI and they have a nice database, and you can start analyzing it. See if you can find new proteins, identify a function of existing proteins, stuff like that. And of course, I urge you everyone to go to your local biohacking group and join up with the central mailing list and just start talking to the rest of the community if you are interested in biology. That's it for me, so if anyone has any questions.
Great talk, thank you very much. If there are any questions, please line up here at this microphone or hold your hand up so I will come to you. No questions? Are there any questions from the mighty internet? Ah, there's one.
What about development evolution, Evo? There is some work going on, but it's quite quiet around this topic. When you look, since this guy told that in 5 years he will give everyone a dino chicken who wants to have one, and I'm still waiting for this dino chicken to happen. What about these biohacking groups? Are you also interested in this kind of topic, this development, evolution stuff?
Can you repeat what the box that you're talking about?
What?
This, you say this guy, he had this, he promised you, I didn't hear.
It was some documentation in Discovery Channel lately about this topic, development evolution, and it was about I'm not studying biology, I'm studying electrotechnics and this stuff, but it was about the states in what is it in English, if this thing in the egg develops to higher stages. So, and first, these little things have tails. Also, human has some fish stuff on him, which develop. Well, anyway, and if you stop the destruction of a tail, even a chicken keeps his tail like a dinosaur.
Yeah.
And they managed to make a chicken with teeth and a tail.
Yeah.
And they're working on keeping this chicken from losing its tail and its teeth and so on before it hatches. And are you also interested in such stuff, or are you only interested in bacteria? That's the question.
So okay, so you're talking about the more macro stuff. Yeah, so most of the stuff I talked about here is microorganisms, and the reason I talked about microorganisms was because they're very easy to genetically engineer, but microorganisms are also being worked on mostly in real science labs and not in backyard biology labs, because they're fairly difficult. It takes a long time to do anything, so there are even more ethical issues involved when you're working with something with a brain and regulations around it. I'm sure people are very interested, but the barrier to entry to working with this kind of stuff is just too difficult. And we're really just starting out in biological communities. And there's so much you can do with the microbiological stuff that I guess we're just taking the low-hanging fruit first.
We have one more question from the audience.
Hi there. I was wondering whether you have done any work to investigate or maybe model the robustness of genetic
I'm having a
modification.
lot of trouble hearing you. Can you?
What?
I can't
Louder?
I can't really hear you.
Okay. I was wondering whether you've done any work towards modeling or understanding robustness of genetic modifications.
Modeling or?
Robustness, whether the organism loses the function after a certain number of generations or under certain circumstances.
Sorry. Robustness. Oh, okay. So you're talking about evolutionary robustness?
Yeah.
Okay. Yeah. So I didn't really talk about evolutionary robustness, but that's also a big problem here because once you engineer these circuits into biology, if they're producing some chemical that's not making it easier for them to live, then evolution will take its course and it'll kill off the ones that are producing it, and ones that figure out how not to produce it will stay alive. So there's a lot you can do to try to prevent this kind of stuff from happening. And I don't think we're at the point in do-it-yourself biology where anyone's really worried about this, because mostly it's one-shot experiments in a lab. So it's mostly something you worry about if you run experiments for a long time at large scales. But as we build more complex circuits, and run them for longer, it'll become a bigger problem. And we'll have to look at how to avoid this kind of stuff. So you can use specifically engineered organisms that tend to not mutate as much and move around their DNA as much, So you have less of a chance of getting rid of the stuff you put into them. You can also try to give them an advantage, of course, but that's not always possible. Grow them on something that makes sure they die off if they stop doing what they're supposed to do. There are different strategies, and a lot of work is going into this kind of stuff in industry, because you really don't want your big industrial reactor with 1,000 or 10,000 liters of something valuable to stop producing, or because of evolution.
With the license, it was no harmful uses. Is that up for definition? Has there been any discussion about that? Or
No what uses?
No harmful uses.
No harmful uses. Yeah, no, there's no definition. It just says no harmful uses. So that's up for discussion. It's still a draft license. So if you feel like discussing it, go ahead on the mailing list. I'm sure they'll be happy to have you.
Okay, thanks.
Yeah.
Hi, you showed us geoelectrophoresis device.
Yeah.
Who did license you to use ethidium bromide or equivalent DNA dye? And how many companies are actually willing to sell it to you without you being a scientific institution?
How many governments are willing to sell you this?
Companies, I would rather
Companies,
say.
yeah. So
Those ethidium bromide and the other DNA dyes are quite interesting chemicals with very interesting properties.
yeah. Okay. So question is about the electrophoresis device that can separate out DNA and proteins of different sizes. And in this device, you use this thing called ethidium bromide, I think, ethyl bromide, and that's a coloring agent that attaches inside of DNA and makes it light up under a black light. It makes it really easy to see where the DNA is. The problem is it's really dangerous. It's cancerogenic. You don't want to get it on your skin because it goes into your DNA and causes damage. So there are a lot of different alternatives. There's something called cyber safe that you can get that is fairly easy to work with and not so problematic. You don't need to worry too much about getting cancer with that stuff. I know you can get companies to ship that kind of stuff to you in the US, but I'm not sure about the European Union. I haven't tried getting anyone to ship that to me. But there are definitely alternatives that you can use that you can get a hold of depending on your country.
We are using ethidium, which is quite, well, it works just fine.
Okay.
That's pretty much the question. You answered it already. Thank you.
Wouldn't it be a very useful general approach in modification of microbiological organisms to just rely on modifying or extending the plasmid instead of manipulating the original DNA?
You're talking about using the existing proteins instead of trying to build our own?
The proteins are expressed relying on the information of either the DNA or the plasmid. And if you only modify the plasmid, which is a lot easier to accomplish than the DNA, I think wouldn't that accelerate the development of useful uses of microbiological organisms?
I'm still not sure you're talking about the difference between using existing genes.
No, building them from the ground up. Also, not using genes that are known, what they express, but actually just, well, I wouldn't say randomly assembling nucleotide pairs, because that doesn't really make much sense.
So you're talking about using selection processes. And,
yes.
Yeah. Okay. Yeah. So I talk about rational design of microorganisms and genetic systems. The rational design of putting genes together and then having some kind of expected function and then checking if you get it. That's the way we build other systems. But in biology, of course, we can use a different process. We can use evolution to our advantage. So you can put in some of the genes you're interested in, and you can make lots of different combinations, and then you can use selection to try to weed out the ones that didn't go so well. You can try to use mutation, so you just try to mutate to get what you need. If you just mutate with nothing else, and you want something really advanced, let's say you want a cancer-killing machine, it needs a lot of different parts, so it's very difficult to get that just randomly mutating. You probably have to wait a billion years or more. But if you have something that's very simple, that's like making an E. coli resistant to some kind of chemical, then you can definitely get that functionality just by mutating. There are some really powerful approaches. You should look up something called MAGE, multiplex automated genome evolution, where they combine evolution with rational design. So they take a lot of different components and they put them in the order that they kind of think they should be, but then they make combinations that have little variations, and they can make a lot of different combinations, millions of different combinations at the same time using this very smart process, and then they just select for the ones that work, and they find that, and then they start looking at how does the DNA look there? Can we go in and further rationally design? So you can get evolution to help you out.
Okay, that's a nice idea. Thank you.
We have one more question.
Hi. I was just wishing to ask, we saw many biolabs around and some pictures of them. What about the environment? Is it possible to build something like such in a normal hackerspace cellar, or there are some requirements, and or the devices are enough isolated from the environment? This kind of.
Oh, okay, yeah. So how much do you need? How much equipment and isolation do you need to grow this kind of stuff? Well, if you have a very dirty lab and where a lot of people go there, and you will have more problems with contamination. You definitely can do it in some place where it's not very clean, but generally you should just get a really good lab bench, and you should put it in an area where there's not a lot of people moving about without lab coats on, and just very basic stuff, really. You don't need a lot. The law doesn't require you to have a lot, even in the European Union. You need some surfaces that are easy to clean and can be cleaned very well. And you need a door that locks, basically. And if you want to do something really advanced, like all the stuff I've talked about here is biosafety level 1 stuff. And that's the certification that we're kind of aiming at to getting first go. But if you want to do something that's a bit more dangerous or more advanced, sometimes you need biosafety level 2, 3, or 4, which is used for dealing with stuff that has the potential to harm humans. And of course, there's some legal requirements about the isolation, but it's not really extremely difficult to get the isolation for these kinds of bacteria.
Was more on the practical side, just to start with the basic stuff, which kind of contamination should be avoided, and yeah, normal praxis, let's say.
Well, yeah, wear gloves, wear a lab coat, don't spit in your petri dishes. It's really not especially difficult. You always get some contamination. If you're getting a lot of contamination and nothing works, then maybe you should move to a different area or clean the area. But it's not, the requirements are not that great. Thanks.
I'm going to try to add something to what you just said, because in Germany and throughout most of Europe, you have to set up a lab that fulfills the legal requirements, but you also need to have 2 people who have a degree in molecular biology or medicine or biology and 3 years of work experience in a Gentech lab.
Okay.
And the biosafety training. So the bar is actually quite high for getting NS1 lab.
Yeah.
And you do have some leeway, but not much there. And also lots of companies will not actually ship stuff to you.
Yeah, so there's a lot of difference between the different countries. And I know also in Denmark it can be very difficult to get the certification. You also need some people who have the correct training. And some countries need everyone to have the correct training. And also, yeah, you do need stuff like that in some countries. Most of it you can probably get used, and you can still build a lab that lives up to the requirements. But yeah, the bar is pretty high. I agree. The problem is that the thing is that most of the difficult requirements are the ones about training and the people who have to be responsible and write logbooks and the documentation you have to fill out and all the processes you have to go through. The actual sanitation and keeping all the surfaces clean and keeping it locked away seems to be, to me at least, the easiest part of the process.
Okay, we do have one very last question on biohacking.
You mentioned that you had some kind of brainstorming process for the ethics for biohackers. Is there any specific outcome yet besides don't be evil?
Well, yeah, there is a draft version. I'm sure you can find it online on doyourselfbio.org on the mailing list. There's been a lot of discussion. There was sent out a link to a document. I'm pretty sure it's published now, but I have to check. It might not be public yet. There was a discussion after the two workshops, and I'm pretty sure they've published the results now. There's still preliminary results and there's still discussion, but if you want to have a look and go, I don't have them unfortunately right here. Mostly with stuff like respect your environment, or we'll respect our environment, and yeah, you say don't be evil and we'll be responsible. There's also some wording about transparency and openness. Mostly it's what you would expect of free software people working in biology.
Thank you very much.
Okay, that's it for this talk. Please thank Mr. Biohacking. We'll just call him Mr. Biohacking. Thank you very much. As you leave this room, please have a look under your seats, right and left at your seats. If you see anything that isn't supposed to be there, take it out with you. We have trash bags right there. Keep this place nice and cozy for the talks to come.